first out of the way, I have no disclosure of interest because this stuff is so theoretical that it's of no use to anyone. But it might still be fun. So I'm pretty much an outsider. I've been doing DNA research in 2001. I'm very happy that you invited me along here. But I'm not some kind of weirdo. So I'm not going to tell you here some weird theory on how DNA works. All of this stuff is pure classical science. There's nothing new in here. It's only that I bring it all together. I may be an outsider, but I've managed to fool some real scientists into working with me. And I got this work published in Nature Scientific Data. So it should be somewhat real. This is, of course, well known. The first parity rule of DNA, for every C we have a G. And this is not in any way really interesting. Much more interesting is that in 1968, Ernst Chargaff discovered that there is a parity rule for each strand as well. So if you take one strand of DNA, you will find equal amounts of C's and G's in there, more or less, and equal amounts of A and T in there. And this is far less widely known, and that's also partially because we don't know why this happens. But it does very manifestly happen. So this is for over 20,000 microbial chromosomes. So this is, well, it's actually bacterial chromosomes, but also archaea, because they're also part of the ecosystem. And you see here the distribution on the percentage of G compared to the total amount of GC. So here you see that the distribution is nearly exactly 50-50 G and C for every strand. And this graph is so neat and so nice because it goes from 49% to 51%. So this is really tight. And some people see in this some kind of miracle, some kind of coordination. Other people say, no, it's just random processes and it happens. And we honestly don't quite know. And it gets a bit weirder. Turns out that if you take a k-mer, like for 6, for example, and you reverse complement it, that you find that the percentage of that k-mer reverse complemented is the same. So this is somewhere. So if you take ACC, GTC, and you measure how much of that do we see in all bacteria, and you reverse complement it, you will find almost the exact same concentration. And this is a graph that shows the correspondence of this for 32 random 6-mers or hexamers. And either this is some kind of miracle or not, and no one really knows. And there are people that say, look, this is just statistics and it happens by accident. And other people say, no, there must be some biological process behind it. And you are sort of free to enter this debate and write a paper that simulates that it's all just random processes. So recapitulating. There is an equal percentage of G and C in all bacteria and all archaea, which is weird, but now let's see where that gets us. In 1996, Lobry in France discovered that this was not actually true. If you look at the circular chromosome of a bacterium, you find that the first half of the chromosome has an excess of G over C. And the second half has a deficit of G over C. So over the whole circular chromosome there are equal amounts of G's and C's, but in the beginning there's a bit more G, and at the end there's a bit more C, where we define beginning as the origin of replication. And this ends at the terminus. Is this a huge effect? Well, it's 2.5%, but it's robust. You will almost not find a bacterium that does not have this. So what does it look like? Well, you don't see it in archaea, which is actually sort of a fun thing, but it's because this is one of the many ways in which you can say that the archaea are really doing different things. What does it look like? I have to admit, this is the prettiest graph that you can make. This is the model organism for GC skew. And what you see here is on the x-axis, we travel over the chromosome. So we start at the 0 point, the origin of replication. In the middle, we encounter the terminus, and then we go on again to the end of the chromosome. So this is a 3.5 million base pair chromosome. And what you see, what we plot here, is we count the excess of G's. So we travel over the chromosome. At the beginning, we have no excess of G's over C's. So this is a cumulative graph. And you see that as we enter the middle of the chromosome, we have an excess of maybe 90,000 Gs. And from that point on, that excess gets sort of compensated, and at the end, we're sort of back down. We're not quite back down. So there's actually, overall, this bacterium has slightly more Gs than Cs. This is very robust. This is just the prettiest graph, but even for other bacteria, it still almost looks like this. The reason I showed this one, this is probably the straightest angle you're ever going to see in a biology graph. Nothing is this pointy in biology, but apparently here it is. So why does this happen even? Now, we know that when bacteria replicate, they do so doing two forks. And that means that one half of the chromosome is copied in one direction and the other half is copied in the other direction, because there are two replication forks traveling in opposite directions. This goes for almost all bacteria. Doesn't go for the archaea, and also doesn't go for eukaryotes. Meanwhile, and there is never an excuse not to show this movie, we of course know that we have the leading strand, which gets copied very straightforwardly, and the lagging strand that has all this Okazaki stuff going on. That means there is a profound difference between how one strand of the bacterium gets copied compared to how the other one gets copied. Because we have these two replication forks that meet each other in the middle, one half of the chromosome is copied in the boring direction, and the other half is copied with all the Okazaki things. That is a reason why you might have an asymmetry between the one half of the bacterium and the other one. So people thought, well, so this is what that looks like. As we move over the circular chromosome, we first build up an excess and then we break it down again. This is a more typical graph. So I showed you the most pretty one before. This is sort of a random bacterium where you still see, it's still a pretty neat and pointy line. So why does this happen? And the spoiler is we don't really know. So just tell you that already. There is deamination. So once we have one half of the chromosome that just hangs in the wind there a bit while on the lagging strand, and there are more chances for deamination going on there. So it might be that you have C's turning into U's, turning into T's, and that could be. Meanwhile, genes, for reasons which we also do not understand, prefer to live in the positive sense on the leading strand. So genes prefer to actually be on the part that is copied straightforwardly in their natural direction. But I mean, I would want to be there if I was a gene, but I don't know how it knows. So that's one theory, is also that since that's the part that gets transcribed a lot, it also gets more repairs in it. That's one other theory. A third one, why this might be happen, is codon bias multiplied by strand bias. So what does that mean? If the codon bias of a bacterium is such that genes just have more Gs than Cs, and you combine that with the fact that genes like to huddle on the leading strand, then that combination could get you an excess of Gs over Cs on one half of the chromosome. Could be. Then there is the third codon position, which can change rather arbitrarily, and it might be under some kind of selection pressure. Well, the spoiler is there have been lots of papers written about this, maybe like 20, which is not enough. And none of these comes to a complete conclusion. And that's also why I started my own work, my own research, because it was like, no one knows why this works. I'm a computer guy. What can I do? I can make a database. That's what I do. So I took my computer programming skills. I downloaded all 32,000 chromosomes I could find on the NCBI website of all bacteria, archaea, and plasmids. And I thought, let's calculate the level of this skew for all of them and see what we get. And once I was at it, let's also split that up over the codon positions. And also, interestingly, if you have G over C skew, you might also have T over A skew, because it's sort of the same thing. And it turns out many bacteria do that. So, and there are some nice graphs about that. I also input in the database codon biases, statistics on stop codons, start codons, GC skew, which was a quality assurance thing just to make sure that I actually found the genes all correctly and that worked. And you can download this on skewdb.org. There you can also find this presentation and the paper, so there's no need to try to memorize. You can still take photos if you want. But on SkewDB there's everything. And there's also an easy-to-use Jupyter notebook where you can play around with this. So what does that look like? C difficile, our favorite one. Well, you shouldn't have it, but at least not too much. And here you see that there is the top graph is the GC skew, which we have seen a lot already, which is again pop, pop, it's rather nice. And the bottom graph is the TA skew. So where you can see that on the leading strand there is a deficit of T's. And on the lagging strand it makes up for that again. But the crucial thing here is to see that the GC skew and the TA skew are in opposite directions. This turns out to go for almost all firmicutes, but not for the other bacteria. And this is, for example, Pseudomonas. I did research on this one, so I like this one. Here you can see that they are in the same direction. So when we have an excess of G's, we also have an excess of T's. Just not in the same magnitude, but it's at least in the same direction. So you can see here that there's a sort of big difference between this one, which is in opposite ones, and this is in parallel ones. So which leads us, of course, to the question, is there some kind of interesting pattern there? There is. So this is all 32,000 chromosomes in there, split out by they keep changing the names for these bacteria, but this is the big groups that they used when I started this research. There are new groups now. The orange ones are the firmicutes, and you can see that they are in the sort of bottom two quadrants, which means that the TA skew is opposite to the GC skew. And all kinds of other bacteria have it aligned, and some of them have more of this stuff going on, some of this have less going on. Archaea have none of this stuff going on, because they copy their DNA in lots of places at the same time. Actually, we don't really know how they do that. But from this graph, you can really see they are not doing it like the other bacteria. Interestingly enough, of course, we have a lot of research going on now on if there are sort of archaea which are a bit more bacterial than others. And if you do that, you actually find sort of bacteria that are a bit more archaeal in their patterns and some archaea that are a bit more bacterial in their patterns. So this is one interesting way, again, to settle the debate, is my lovely organism actually more like this or more like that? You can look it up in the SkewDB. Actually, this is from the live viewer, skewdb.org/view, and has an autocomplete, so you can just enter a few letters of the name of an organism and it will show you the graph. And there you can also find some really weird bacteria. So here are 4 of them, and again, you can find them through skewdb.org. And maybe this is one of your favorite organisms, I don't know. I don't recognize these sequence things, neither do you probably. But here you see that there are a bunch of bacteria, and there are hundreds of them in the database, on which one half of the chromosome has very clear GC skew, and the other just not. That is supremely weird, because the mechanism behind it is fundamentally symmetrical. So, I don't know what's going on, but maybe it's interesting to ponder. And the other thing is, why does a bacterium have these 2 replication forks? Well, it's because it's faster. At least that's why we think it is the case. In order for that to work, both of these copying forks need to travel at the same distance. Because it doesn't work if you have one really big replication fork and then one small one. Because that doesn't give you the speed up that it could get you. Well, this is nature. So here are a bunch of bacteria that have said, well, actually, one part of my replication fork is a lot shorter than the other one. This would seem to be highly non-optimal. But on the other hand, nature is pretty good at what it does. So it might have a reason for it. It might also mean that these chromosome sequences are not good. That someone sequenced them, assembled them, and they missed half of it. That's one other role you could use my database for, even if you don't want to delve into obscure GC skew stuff. You can very easily spot that a chromosome is weird. For example, this one. It goes up, goes down, goes up again a bit, goes down a bit. What could be behind this? This could be sloppy DNA assembly. could also be that this chromosome integrated plasmids, which don't know about the local GC skew conditions, so they bring their own GC skew. So this could be a sort of forensic indication that either this was misassembled or this bacterium had some wild adventures in the recent past. Could be. So, some possible uses of this database. It's rather fun to just type in your favorite organism and see if it might be a weirdo. Because you might just have a fridge full of interesting bacteria that could help us answer questions about that DNA replication. It could also be a QA measurement, because if it looks entirely weird and either your assembly is weird or your organism is weird, or maybe you are weird, then could still be. And it's also very interesting if you have some of these sort of interesting new, very hard to culture organisms going along where people are not really sure if it's archaeal or something in between. This can be a tiebreaker. This is a rather fun thing to look at. And of course, the big thing is, it is a sort of glaring omission that we know that this stuff goes on, and we don't really know why we have the same amount of G and C on each strand in the first place. That's not quite clear. We don't really know why that holds for larger k-mers. Could be coincidence, but no one has proven that it's just statistics. So there is something deep and fundamental here that we don't understand. But yeah, it's of no use directly. So it doesn't quite fit in the funding narrative, I'm afraid. And so I spent all my time only just describing the database without interpreting it. This is one slide that I do find a little bit fascinating. This is the all firmisuits, where we had this hypothesis that this skew is actually caused by codon bias multiplied by the strand bias, by which I mean the tendency for genes to huddle on the leading strand. And this is for all firmicides, and you see that actually this hypothesis would lead to the two black lines. And, or actually it would lead to straight lines. And you see for the down, which is TA skew, it says if there is no gene bias or no codon bias, there is also no TA bias. The line goes straight through the origin, which seems to suggest that TA skew is just caused by codon bias and strand bias, which still leaves unexplained why these genes huddle over there, but anyhow. But for the GC skew, you very robustly see that it does not go to zero. That means that if you are looking for an explanation, at least in firmisuits, there is something more going on than just codon bias and strand bias. And this not only holds if you make this graph for all firmicides, but it also holds if you do the graph for an individual firmicide and you plot the local strand bias. So this is a sort of suggestive thing that if you would actually want to make of this, for firmicides we have at least some concrete results. And that ends my presentation.
Very nice presentation. Do we have some questions from the audience? I think we have plenty. Yes, I'll get the floor to you.
Yeah, what about plasmids? First of all, very interesting work.
Thank you.
I really like it, honestly. And what about plasmids?
That's a very good question. The plasmids are clearly marked in the database as being plasmids. So you can sort of separate them in your graphs. It turns out that there is a spectrum. The larger plasmids are like sort of pseudo-chromosomes. They just have the same kind of behavior. But there are plasmids that just don't work like this. So and that's also interesting because that maybe tells you something about their age. Because whatever mechanism you think that could explain this, it probably takes millions of years to act. So yeah, the plasmids are different, and that's why they are clearly marked in the database, so you can do plasmid-only analysis.
Thank you.
So question here? Yeah.
Have you also looked at high GC content genomes?
I have. And there are lots of interesting things come out there. But nothing that immediately says this is a hint. So if you do that, for example, you see that with the high GC content, the start codons become markedly different, which in itself creates a skew. So that is also in the database. And if anyone, by the way, I'm around all day. I have the database with me, and I will be showing it there And I have the Jupyter notebook, so we can do, if you want to plot the GC content versus the high GC, lots of interesting graphs come out. More than I can explain.
Yeah. No, I asked because I work with bacteria, 72%.
Wow, yeah.
Okay.
Any more questions? Well, he's around the whole day, so if
Yeah.
you have still some questions, you can find him.
Thank you very
I
much.
think you can go to the next speaker.